We've been working on this drug resistance population. We call it the TASP study, which is one of the South African populations in South Africa. So I'll just start with a bit of a few slides, a bit of an overview on patterns of HIV drug resistance with a focus on Africa, and then we'll sort of dive into the sub-study, which is what I presented at the resistance meeting a couple of weeks ago. So I'll just take you through a few figures. So unfortunately, because of the COVID pandemic, there's not been a whole lot of output in terms of HIV and antiretroviral therapy resistance over the last 2-ish years, I'd say. So essentially, what we have showing in this figure is the prevalence of NNRTI resistance. All of these individual circles represent one of the a study which made up this paper by Gupta and colleagues. It was published a couple of years back now. Essentially, what it's showing that over time, going back from 2000 up until 2016. There's just a steady increase in the prevalence of NNRTI resistance across all of the different continents. But unfortunately, it seems to be a lot more, particularly in East Africa and South Africa. So if you continue to extrapolate these figures, unfortunately, we see that the prevalence of NNRTI resistance has just continued to increase over time. So here is another figure which looks at the prevalence of tenofovir resistance, and this is in association with thymidine analog mutations, which are those sorts of mutations which are selected for by AZT and D40. And again, you can just see, if you imagine a relatively straight line coming through this, the higher the prevalence of tenofovir resistance, the more people you see with these thymidine analog mutations. And then here's one from Mark Seidner, who's one of our colleagues at ARI, who recently, well, I think it was year before last now, was running a trial called the ADVANCE trial, which essentially showed that there was a high proportion of people with pre-treatment drug resistance. And the vast majority of these mutations, which made up that pre-treatment drug resistance, were, of course, NRTIs and NNRTIs. So from these three figures, essentially, we can see that there's ever increasing proportion of people with NNRTI resistance, in particular to efavirenz from most of these studies. So here is another figure which looks at spreads of different HIV-1 subtypes and rates of, again, tenofovir mutations. The only take home I have from this is the rates of tenofovir mutations particularly seem to be highest, again, in East Africa, South Africa, and Western Central Africa. You can see here the ones that I've circled, specifically because we work on a lot of drug resistance in South Africa. So Sikilili, who is one of our PANGEA collaborators as well, published this study in PLOS1, which was looking at the rates of antiretroviral drug resistance in the 2017 National HIV Household Study, which was conducted in South Africa. And again, he showed the same sorts of things that we're seeing. There's a large proportion of people with NNRTI resistance. So this here is single class regimen resistance. And then over here we have NNRTI and NRTI, which is dual class drug resistance. And then he also showed that there's a small but slowly increasing proportion of people that also have protease inhibitor resistance or triple class resistance. Now, this one is the last figure I'll show you from recent literature. This is a publication which was showing people on boosted protease inhibitor regimens. So we call that second line regimens, essentially. And this is showing you that there's a high proportion of people, again, with these characteristic NRTI and NNRTI mutations. And they found what we normally find when we look at drug resistance across Africa, which is there's a high proportion of people with M184V mutations, which is a characteristic NRTI mutation, and K103N, which is a characteristic NNRTI resistance mutation. Now, what's interesting, particularly about this figure, is all of these people on their boosted protease inhibitor regimens, we're starting to see people with 10, 15%, well, 10 or 15% of people on these regimens with resistance to boosted protease inhibitors, so lopinavir and ATV in adults under the age of 16 years. So the take home from this slide really is that we know, and we have known for some time, that there's a high proportion of people with NNRTI and NRTI resistance. But what's now becoming more troubling is we're starting to see a rise in resistance to protease inhibitors. So what we've been working on as part of this TASP sub-study, I guess, is essentially trying to figure out what's happening to people when they're failing second-line regimens which rely on these boosted protease inhibitors. So in South Africa in particular, we all know that there's been this new switch and new rollout to dolutegravir-based regimens. However, it's likely that in the very near future, at least many South Africans will continue to rely on boosted protease inhibitors as part of their regimens. So it makes sense for us to continue to investigate what's happening and tracking protease inhibitor mutations. So this leads us to a slightly revised title from what I showed at the resistance meeting last week. So essentially what we've been looking at over the past, I'd say 2.5 years since I started working for PANGEA, but more intensively recently, is competition between different viral populations whilst they're under non-suppressive ART. And we have a few questions which we've been specifically trying to answer. And some of these include, why are people not suppressing on what should be extremely effective treatments? Is it that there's different aspects of resistance at play? Is it that there's different populations? And then we also want to know, why is it that there seems to be a large absence of major protease inhibitor or drug resistance mutations when people are all taking these therapies? So for anyone that doesn't know, I think you should by now if you've watched any of my previous talks, the TASP study was an ANRS study, which was conducted by predominantly the French government. Was conducted in KwaZulu-Natal province, which is this big area you can see where my laser pointer is, and the epicenter, which I shouldn't probably call that anymore, going from our previous talks, is around this district here. This is where most of the samples were collected from. So the TASP trial was designed to study long-term impacts of people taking ARTs. It was a population-based cohort, which essentially had two arms. The control arm was where people presented to the clinics, and if they were diagnosed with HIV, people in the control arm adhered to the current prevailing South African guidelines, which was to have a CD4 test. If they had to wait for a certain CD4 level before they were given ART, and that was compared to the intervention arm, which immediately placed people on ART. And the idea was that it would essentially decrease population level of HIV incidence, but unfortunately that the TASP trial was actually one of these universal test and treat strategies that was deemed a failure. And it was a failure in the sense that there was actually no significant difference in the control versus the intervention arms. And there's been various different thoughts over the years as to why that was. And I think the prevailing theory is that a lot of people were just lost to care. So a lot of the infections were coming from outside of the study area and then infecting people within and then traveling back out again. So the participants that we followed closely in this sub-trial were a subset of 8 chronically infected patients. And from these, we have roughly 6 monthly samples, which were followed longitudinally for up to about 3 years, I think is our longest one. So we analyzed basically 44 sequences, which represent 44 longitudinal samples. And all of these people are characterized by numerous suppression and, well, partial suppression, I'd say, because they never suppressed to less than 50 copies per mil, partial suppression and rebound events. So prior to engaging with the study, most people would be taking this first-line regimen, which consists of 2 NRTIs and 1 NNRTI. And then when they joined the TASP trial, they would have been switched to this regimen, which is 2 NRTIs and a protease inhibitor, which in most cases was boosted lopinavir. And then, as most of you know, South African government is now recommending dolutegravir-based first-line regimens. But as I mentioned, majority of people will still remain on this second-line regimen for some time. So what we first wanted to do was essentially investigate why most of these people were failing. So some of the traditional arguments are that they are not adherent or something like that. So we started to investigate a bit more closely. So what we have here is a multi-dimensional scaling plot, which was produced with the help of one of my co-authors on the paper, Oscar, who's at UCL. So what we did was we looked at the consensus sequences for each of these patients. We put all the time points together and then calculated the average pairwise distance between those sequences, and then essentially analyzed the clustering patterns to see if there was a large proportion of diversity. And what you can see from here is each of these green dots represents one time point, and they're colored according to the patient number. So most of them do cluster quite closely together, but some are a little bit more diverse than others, suggesting that there could be some sort of superinfection or lots of different wild swings in sequence due to a host of factors. So if we zoom in here a little bit, you can see particularly with this, this patient, which is patient 28545, two of the sequences are up here, and then three of them are a little bit down here. And again, with this other patient, 22828, four of the time points cluster quite nicely together, and then two of them are significantly further away, which suggests that there's a high degree of diversity going on. So in order to corroborate this, we used a program which was developed by Richard Goldstein at UCL. This basically examines nucleotide diversity. And rather than looking at consensus sequences, this actually takes a BAM file as an input. So it uses all of the sequencing data. And another benefit is that it doesn't actually require any sort of pre-filtering to remove sites with low read depth. So again, you can see that there's some significant diversity between different time points. So if, say, for instance, in this patient here, each of their time points are quite significantly different. So using these two methods, what we essentially did is determine some sort of cutoff for what we called significant diversity. And then we tried to decide if we wanted to computationally reconstruct haplotypes. So we decided that from these two multidimensional scaling plots and this measure of nucleotide diversity, we were convinced that there were different haplotypes within each patient. So another program, which was designed by Richard Goldstein, is called Harald. So this is the program that we use to build haplotypes or to reconstruct them in silico. There's a whole paper on this, which is written by Winita Pang. It's currently on bioRxiv. I think it's been submitted to a journal, which is the validation paper showing how the method works. But very briefly, essentially what you do is you have a person who's infected with a virus. You obviously take a sample, you extract your viral RNA or your DNA if you want to look at DNA virus. You get some sequences, and then what Harald does is essentially creates a set of haplotypes whose variant combinations most optimally account for the observed frequencies in the sequence. So essentially, the output of Harald is a set of haplotypes that reflect the highest log likelihood of the observed frequencies. So there's two different steps to that method. You can read about it more in the paper if you're interested in reconstructing haplotypes downstream for resistance analysis or whatever. But the first step assumes that there's a set number of haplotypes between different time points, and then there's a refinement step afterwards, which relaxes that assumption essentially, and gives you the number of haplotypes it thinks is most optimal per time point. And Harald was specifically designed to take longitudinal sampling into account. So you're asking me, okay, you've done haplotypes. What was the point of looking at haplotypes? So here's quite a nice figure, which is from Ravi, essentially looking at selective pressures of therapy. So when a person is first infected with HIV, for example, they'll have a particularly high viral load. something in the region of four times 10 to the 10 viruses per milliliter of blood. When the treatment begins, you'd expect that if a person is completely adherent to that regimen, the viral load will slowly drop off. You'll start to destroy drug-susceptible populations. But unfortunately, when adherence is not as good as it should be, you'll always start to get a smaller population that will begin to develop resistance mutations. And then unfortunately, due to a number of reasons, which are listed here, the regimen that you're currently on will destroy this drug-susceptible population, and it will leave a smaller drug-resistant population, which will then slowly increase after replication over time. So what we did was, for each of the 8 patients that I just mentioned in the sub-study, we reconstructed viral haplotypes, and each of them are shown here in a maximum likelihood tree. Each color is a different patient, and what we'll do is we'll drill down into 3 of these shortly. But what we can tell from this tree is that there's ongoing competition for viral emergence. There's not a particularly distinctive pattern, shall I say? So each of the time points, so say for instance, this patient here had, say for instance, 4 longitudinal time points. They're all intermingled together, which suggests that there's been some sort of dynamic shifting in populations between those time points. So what I'll do is I'll take you through the analysis that we've done for 3 of these patients. So the output of Harald will essentially give us something which looks like this. So here you have the patient number, and then you have the time point. So for this patient, there was 1, 2, 3, 4, 5, 6 time points. And then this is the haplotype number. So with this, we can also tell which are the majority haplotypes, which are the minority haplotypes. It gives you, Harald's output gives you quite detailed information regarding each of these. So here you can see that at time point 1, this particular patient had been on a first-line regimen for, I think it was just over 6 years, and they'd been failing it for what's presumed to be some time, as they have these classic mutations, M184V, which are selected by 3TC and FTC, and then you have these characteristic N and RTI mutations, which are all selected for by efavirenz and nebirovine, which is correspondent to what the patient was on for quite some time. So this arrow here is where the patient, at time point 1, the patient had been switched to their second-line regimen, and then by time point 2, the patient had been on their second-line regimen for some time after this time point. So you can see that there's been a complete loss of M184V because there's now been no specific selection by lamivudine or emtricitabine, but they've persisted with this K103N mutation. So variable adherence over time is what's leading to competition in populations. So here is a viral load plot, and thank you very much to Andarash for making the original one of these. We've tweaked them since. So essentially what this shows is in the left-hand column, you can see the regimen that the patient was taking and for how long. And then this first time point, which is represented by this line, reflects when the patient was switched to their second-line regimen as part of the TASP trial. And this is the regimen that they were taking. And then you can see these dotted lines represent the perceived change in viral load over time. So each time point viral load was taken, and viral load was calculated, and the plasma concentration of drug was measured by HPLC, and that's reflected by these numbers here. So what we do is we underlay this with a variant change plot. So here you can see these dark or bolder lines. These all represent major drug resistance mutations, which are according to either IAS or, say, for instance, the Stanford HIV-DB. So these are represented by the bolder lines, and then underneath you can see a series of sort of background non-synonymous mutations. So each of these lines represents a different variant, and what we do is we track them over time to try and discern what's happening underneath, essentially. So here in this patient, you can see that they have, at the first time point, they have these characteristic first-line regimen mutations. So K103N in green, M184V in red, and P225H in blue. After they've been on their therapy for about 8 months, you can see that there's been a complete loss of P225H, which is shown by this dark blue line. And then by months 15 to 16, this patient had a partial suppression. So they suppressed from 5 log right the way down to, I think it was about 700 copies per mil. And then what we can see is that there's been a preferential suppression of those NRTI sensitive viruses, which has actually led M184V to increase in prevalence. So the populations containing M184V have begun to increase over time. So then we look across, and then by month 25, because this patient has been largely non-adherent and has also begun to rebound after month 21, we can see that because there's no particular selective pressure, P225 has increased in prevalence, in frequency, along with M184V. And we think this is largely due to the fact that there's some sort of linkage between these two mutations on a haplotype. So what we do is we link this graph to this one here. So in the paper, we present all of these figures together so you can see a complete picture, but unfortunately there's not enough space on slides. So it's a little bit harder to see, but hopefully you can try and string everything together as we go. So up here, we give an overview of what's happening in the patient, and then we look at non-synonymous changes. And then immediately below that, we also consider the synonymous mutations because we have whole genome sequencing, so it makes sense to look at the whole genome. So we have these characteristic chevroning patterns, which we Josh Herbeck mentioned that he'd also seen previously and published a paper similarly on. So what we say these chevroning patterns are essentially soft selective sweeps happening in the background of hitchhiking on the back of RT drug resistance mutations, essentially. So what's quite interesting is between months 8 and 25, the variant frequencies of these synonymous mutations only seem to differ between sort of 20 and 60%, which is kind of suggestive of ongoing competition. However, this is quite in quite interesting contrast with the first few months of treatment, where you have these sort of these variants decreasing from, say, for instance, close to 100% all the way down to less than 5%, which is suggestive of complete replacement by a more dominant haplotype. So what we do is we look at all of these haplotypes and we construct a maximum likelihood tree of these. So here you can see a maximum likelihood tree. I've omitted the bootstrap value for now. It'll become clear why a little bit later. But essentially, yeah, maximum likelihood tree. And then we have each of the colored tips represents the time point. So these are all longitudinal samples. And then we have the tip size, which represents if it's a majority or a minority haplotype. And we have different definitions of majority or minority depending on how many haplotypes there are or what frequencies they're at. So to the right of that, you can see a heat map, and the heat map just basically displays which drug-resistant mutation accompanies which haplotype. So in this particular phylogeny, we can see that there's a distinct cladal structure. So we have, say, for instance, clade A up here and clade B, which largely splits two different populations of haplotypes. There seems to be more haplotypes with RT mutations down in this lower clade B. What's interesting is we can see that these dominant haplotypes quite accurately reflect the variant frequencies that we're seeing in the previous previous variant plot frequencies. So say, for instance, at time point 1, you remember that this patient had all of those characteristic mutations. They had the K103N, they had the M184V, they had the P225H, and that's also reflected here in the majority haplotype, because we can also see that there's competition in the fact that this other haplotype, which was at time point 1, a minority haplotype, did not in fact carry any of these mutations. So we say that the phylogeny is actually consistent with competing populations. So here at time point 5, we can see that a minority haplotype only carries 2 of the 4 mutations. It carries the K103N and the M184V, whereas the majority haplotype carried all but K238T. So in this particular patient, we saw what we'd say is a hard sweep between time points 1 and 2, which accounts for the large decreases in drug resistance mutations from close to 100% close to less than 5%. So that's for patient 1. So if you think going forwards that we collect all of these different figures together and then we try and present an overall story. So in our second patient, we can see that this has a slightly different story in the fact that this patient only seemed to have one characteristic drug-resistant mutation at high frequency throughout the following period. They didn't ever seem to develop other mutations, which was quite interesting, we thought. This patient is largely non-adherent, as you can see from the low drug levels, which are measured in plasma. And then again, what we can see here is evidence of hard selective sweeps in this, what we call chevroning pattern. So I forgot to mention, sorry, the different colors represent the different genome position. So anything that's colored in blue is largely an envelope. Anything covered in greeny yellow is mostly in pol, and then anything colored in red is mostly GAG. So in the absence of strong selective pressure from drugs, we suggest that these large changes are actually due to either neutralizing antibodies or from T-cell immune pressure. And also what was also interesting about this patient is most of these envelope changes appear to be in GP41, which is actually a position which Professor Eric Hunter suggests are associated with people failing protease inhibitors. So then again, if we switch over to the synonymous mutations, we can see rather interestingly that there's this same chevroning pattern as occurring in the previous slide. The only difference is now, though, that rather than being an envelope, the majority, or certainly more of these, are actually in either GAG or pol. So the idea we have behind this is that the GAG-pol function is vital, but it's not particularly tolerant of non-synonymous changes. And then we also noted that capsid is actually more conserved than matrix, P6 not so much. And then here we have the tree structure of all the haplotypes. Not particularly interesting tree because there's only one drug resistance mutation. The only thing we can really garner from this is that some of the more minority haplotypes are also not carrying the drug resistance mutations. And then finally, we move on to our third patient, which was arguably the most interesting because A, we had the most number of time points to follow, and B, this person was slightly more adherent to their drug regimen and also had these big shifts in drug resistant mutations. So we can see that by month 10, this patient was actually the first we saw that was actually taking their boosted protease inhibitor and one of the NRTIs. But then unfortunately, as they progress, viral load slowly creeps up because they seem to be only taking one of their drugs. So they're essentially on a 3TC monotherapy here. What's really interesting, or most interesting about this patient is we seem to have this antagonism going on. So the antagonism seems to be between K65R, which is a tenofovir mutation, and these two thymidine analog mutations. So we argue that either there's some sort of recombination occurring here from an archived virus, because there's really no selective drug pressure to have these two TAMs appearing from nowhere, because the patient's actually not been on D14 now for over 2 years. And then again, we move over to synonymous mutations, and really there's just a huge hodgepodge of things going on here. So I'd argue this is not the most informative graph of this patient, but there's just 2 points down here that we were discussing, is that either recombination is responsible for some sort of soft sweeps, or there could be some sort of dissociation of variants relative to those drug resistance mutations, which are both increasing and decreasing. And then again, we have the phylogeny for the haplotypes here. This one was, again, one of the first we saw where some of the time points actually clustered by themselves, which is suggestive of a reduction in diversity. So we can see here that time point 2, they all cluster together. Time point 3 all seem to cluster together. So it could be from, like I said, it could be recombination occurring here, or it could be that from time point 3, we've had virus emerge from some sort of cellular reservoir. Also because it's the first time we see the TAM mutant coming up. So largely, we treat this patient as quite an accurate validation of the Harald method, because it seems to quite accurately show that on the background of antagonism, we never see some of these mutations occurring together. So after constructing haplotypes and looking at different phylogenies, we're actually working with Darren Martin now at the University of Cape Town to look at the role of recombination and the role it plays in haplotypes. So obviously we know that constructing phylogenies, maximum likelihood phylogenies, they don't actually consider recombination. And we obviously know that HIV-1 does have a high degree of recombination. So what we're trying to do is essentially determine recombination breakpoints within haplotypes to try and better understand the competition that's going on. And like I said, in association with Darren, we'll essentially use these recombination breakpoints to build better phylogenies, which also take into account recombination. So this is just a very rough tree that he provided to us just yesterday. We're waiting to talk to him a little bit more about this. And then what we're also looking at in association with Oscar at UCL is the role of selection in these populations. So what I have here is a plot of DNDS, so rate of non-synonymous versus rate of synonymous substitutions on the y-axis, and then on the x-axis we have gene by gene nucleotide position. So this first plot here shows GAG. And what we have is essentially a red dotted line which shows that the rate of non-synonymous and synonymous change is 1, which effectively shows neutral evolution. The further it is to the north, it would indicate positive selection. And then the further it gets to the south of this line would show positions which are under strong negative selection. So these blue sites are ones which I've basically scouted literature for, and they've all been shown to have some sort of association with virological failure in patients who are on boosted protease inhibitors. So that would fit our patients quite nicely. And interesting idea is that some of these are actually linked. So we have, say, for instance, mutations in GAG. So say, for instance, this L75R, which we can see is positively selected over here, seems to be linked to one of these protease mutations, M46I, which is selected for by boosted lopinavir. So this is one of the mutations in one of our patients. So as I mentioned, we did a gene by gene. So I showed you GAG just now, and now here's POL. So in this left-hand plot, we can see reverse transcriptase, and then in middle protease. What's quite interesting is we seem to see that a lot of these drug-resistant mutations seem to be under quite strong negative selection, with some of these lesser ones up here, except P225H, which is quite prevalent in most of our patients, at least at first time points, seems to be under positive selection. In protease, not really much going on. And then when we look at integrase, there's, say, for instance, quite a lot of neutral selection happening of DRM sites in particular, but most of the other positions seem to be under quite strong negative selection. And then finally, we look at ONV, which is basically a huge hodgepodge of largely neutral evolution. What's interesting here is in this lower right-hand side, we can see that a lot of these sites seem to be under strong negative selection. And if you remember back from one of my earlier slides, I mentioned that Eric Hunter suggested that mutations in envelope had some association with PI failure. And we see some of these sites which Eric Hunter has described. And the importance of this is that GP41 is obviously very important for host cell infection and entry into cells it's infecting. So the take-homes of a lot of the work that we've been doing so far are these. So essentially, examining intra-host evolution during stable viral load reveals complex changes So typically when we used to use Sanger sequencing, we could see drug resistant mutations, we could tell you if it was there or not. But now with NGS, we can dig a lot deeper essentially and see really what's happening. We've also shown that there's various selective sweeps going on within our patients, and that we've seen that non-synonymous changes can be constrained in more conserved genes like POL. And then that's why we say that synonymous changes, examining them is also quite useful to do. And then lastly, we've shown that haplotype reconstruction has actually given us really good insight into what's really going on in these patients. And we've shown that it's actually quite effective at, well, if not conservatively, it's given us a good idea of the different viral populations in each of our patients. We've actually done some in vitro work using single genome amplification to essentially validate these. And then lastly, we're just looking at the impacts of selection and recombination and what they contribute to competition between different viral populations. So I'll take a deep breath there and thank some people, and then try and answer any questions that anyone might have.